Hi Hires, we are looking at the structure of a variety of proteins that you find inside cells in the DNA and Genome Unit. So ever since you've started biology, you should be aware that cells have particular shapes because they have particular jobs. Their function is totally linked to the shape they have. If they didn't have the shape, then they couldn't do the function, and because they have that particular function, they need to have that particular shape. It is a central core of everything to do with biology. Proteins are the things that actually make things happen inside cells, and so therefore they have exactly the same thing. The shape of a protein is totally linked to the function that protein has, and the function that protein has is going to link to the shape it has to have. Shape and function are constantly linked, and there's the big argument about what comes first, the shape or the function or the shape. It is the chicken and egg thing. So we have another problem in our understanding of what's going on. We know that actually it's not all of your genome that is expressed. It's only a tiny amount. And we also know that there are more different proteins that you have in your cell than the number of genes, which doesn't really help us because we think of a gene being something that codes for a protein. That's fine. If you have 10 genes, you have 10 proteins. That's just not true. So there's now been a real upsurge in understanding and a lot of research trying to understand how we can have one gene giving us lots of different proteins. And the proteins are called the proteons, and so the proteome rather than genome. And so if we've got a genome which we think is around 25,000 genes, how on earth can we get to over 1 million different proteins? And there's loads and loads of different ways that we do it. And luckily there's only one that you need to know about, and that is alternative splicing. All of the rest of them are fascinating, really interesting stuff, and if you go on to do advanced higher, then you'll talk about that in a bit more detail. So splicing we should already know. Splicing is where we remove the introns and leave the coding exons. But what we now know is you could actually have a gene with, say, 10 exons and produce a protein which only used, say, 5 of them. But which 5? You could pick and choose which 5, and so from 10 different exons you've got a huge number of different potentially splices that you could have. So one of our best examples is antibody production, because that's got a section of the antibody which is always the same and then a bit at the top which is different. So the bit that's the same you keep the same coding for, and then you just change the bit at the end. Okay, this is the nicest diagram I could find of this. This is not antibody production, this is a general one. So what we've got here on the original code, so this is your original DNA code, we have exon 1, 2, 3, 4, 5, 6, and then we've also got these pale blue sequences, so these are our introns. So what we'd normally say is, fine, find the promoter, bring in our RNA polymerase, open up a transcription bubble, start reading along, and then peel off our primary transcript. And now all we need to do is just cut out those introns. So if we did that, we would end up with a normal sequence which ran 1, 2, 3, 4, 5, 6 on the exons. But if we do alternative splicing, we could do something like, okay, for this one, for the first one here, what we've done is we have spliced 1, 2 as normal, and then we've cut at the end of 2 and spliced along to the start of 4. And then we've done our normal cut at 4, 5 and our normal cut at 5, 6. So we've ended up with a different mRNA from what we would normally expect. And there's another one. This time, instead of cutting 1 to 2, you've actually cut 1 to 3, and then 3 has skipped all the way to 5, and then 5 to 6, and a different one here. Okay, so from just one gene, that original chromosome gene, we've got lots and lots of different potential mRNA transcripts, mature transcripts, which can go on and be translated. So each of these proteins is going to be a bit different. And this is alternative splicing. What is really essential is that the exon order doesn't change. So it's the same thing as we had with splicing. You can't say, I'm just going to shuffle them and go 1, 3, 2, 5, 6, 4. You can't do that. But you can cut out bits and give you a different end sequence. Finally, once you've made your protein, we now need to fold the protein, and so this is to give it the actual shape that it's going to be able to do its job. And that folding is going to be stabilized by other bonds. Now you're expected to be able to give examples of other bonds that help you fold the chain. So the original bonds are covalent bonds in the peptide bonds. All of those are strong, linking together what's called the primary structure of the protein. Okay, what you then need to do is, once you've folded it, keep that fold in place. So if you've got a chain that you've managed to say, here was my original chain, and now I've got one that goes like that, I need to have something in here that's going to actually help that stick. Okay, so how you can do that, you might be able to use another covalent bond or something called a disulfide bridge, which is very good at connecting together bits of chains or chains together. You might use hydrophobics, so basically a blob of R groups on amino acids that do not like being near water, and so they will kind of clump together. You could have ionic bonds, so that would be a plus and a minus charge helping stick, and you can have hydrogen bonds. Okay, and hydrogen bonds are a particular form of van der Waals force, and if you're doing chemistry, any one of those. So you could say hydrogen bonds, London dispersion forces, dipole-dipole attractions, whatever you like. But if you're not doing chemistry, I wouldn't worry about them at all. I would go for hydrogen bonds and probably ionic because they're reasonably easy to remember. Make sure you've just got a couple of examples in your head that you can give. And then once you've done that, well, we get every possible shape that you could have. And there are three big kind of groups that we put proteins into. That's what I'm giving you in the next 3 slides. Okay, so the first main group we would say is fibrous. So that's basically ropes, big long chains that you have put into, normally it's an alpha helix, so it's a big kind of twisted ribbon shape. And then you kind of rope them together and twist them together and you get strong supportive fibers. So they're really useful if you need to connect things together, like connective tissues in our body will be made of fibrous things. Our hair, you know, keratin and collagen are 2 of the best examples of that. A lot of your proteins, however, do not have long rope-like structures. Instead, they've got, well, a blobby shape. Okay, they are complicated, complex folds that give a very, very specific folding that allows very particular things to stick to them. So that would be your antibodies, this would be your enzymes, this would be hormones, this would be receptors, everything that depends on having a particular, I'm going to say lock and key, but we'll discuss that later in Unit 2, but that idea, okay? And finally, you've got a group that are basically a normal protein, all peptide linked, and then something else stuck into it. So that might be something like the heme group in hemoglobin, it could be a vitamin, it could be other minerals that are attached to it to make it actually do its job. So here's an example of, this is a picture of hemoglobin. So what you've actually got there is 2 alpha chains, so that was a gene code for the alpha chain, and then the beta chains were coded somewhere else. They made 2 of each of these, locked them together, and then each of these at its core has a heme group, which is a metal ion group, the iron bit that gives us the colour. So if we look at this in the round, basically what we're saying is that it's the final pattern of expression of proteins. So how many proteins you produce and which ones you produce and at what levels, that decides what the cell can do. And that will decide for you as a ridiculously complex multicellular organism, everything about you as well. It is the expression of your coding that matters. So that characteristic that you end up with, that is your phenotype. So your genotype is your coding, your phenotype is what actually happens once you've expressed that. And just to add in another twist, the phenotype could also be influenced by environmental factors. So they might be affecting your actual transcription, your translation. It might be affecting how often you can produce things. And finally, you have potentially epigenetic modification, just to add into that as well. A little bit more on that later. And that's us.